Life. It is a phenomenon that surrounds us, a complex web of interactions and processes that we are only beginning to understand. A symphony of complexity orchestrated by an elegant molecule. DNA. This molecule is the cornerstone of all living organisms, a testament to nature's ingenuity. This blueprint of life, residing within almost every cell of our body, dictates our traits, our predispositions, and even our vulnerabilities. It is the instruction manual that guides the development and functioning of every living being. Understanding this intricate code is fundamental to unraveling the mysteries of life, from its evolution to the development of diseases. By decoding DNA, we can gain insights into the very essence of what makes us who we are. Imagine DNA as a vast library, an immense repository of information. Within this library lie countless books, our genes, each containing instructions for building and maintaining our bodies. These books are meticulously organized, each one playing a crucial role in the grand design of life. These instructions are written in a language of 4 chemical letters, A, T, C, and G. These letters form the words and sentences that make up the genetic code. The precise order of these letters determines the function of each gene. Just as the arrangement of letters in a sentence can change its meaning, so too can the sequence of DNA letters alter the function of a gene. Deciphering this genetic language, understanding where genes are located, and determining their sequence are the goals of genetic mapping and sequencing. These processes are akin to translating an ancient manuscript, revealing secrets that have been hidden for millennia. These powerful tools have revolutionized our understanding of life, enabling us to pinpoint genes involved in diseases, trace our ancestry, and even engineer organisms with beneficial traits. The potential applications are vast and transformative. Just as a map guides us through unfamiliar terrain, genetic maps help us navigate the vast landscape of DNA. They provide a visual representation of the genetic terrain, highlighting key features and landmarks. They provide a framework showing us the relative positions of genes along a chromosome, much like landmarks on a road map. This framework is essential for understanding the complex interactions between different genes. Sequencing, on the other hand, reveals the precise order of the genetic letters within a DNA molecule, allowing us to read the language of life itself. This detailed reading is crucial for identifying mutations and understanding genetic disorders. Mapping the genome, like charting the stars, requires observing patterns and relationships. It's a meticulous process that involves understanding the intricate connections between various genetic elements. Early geneticists, lacking the tools to directly analyze DNA, relied on the observable traits passed down through generations. They meticulously recorded these traits, creating detailed family trees and breeding experiments to track how characteristics were inherited. By carefully analyzing the inheritance patterns of these traits, they began to construct genetic maps. These maps were rudimentary, but provided a framework for understanding how traits were linked and passed down. These early maps were based on the principle of genetic linkage. This principle suggested that certain traits were often inherited together because the genes responsible for them were located close to each other on the same chromosome. Genes located close together on a chromosome tend to be inherited together. This proximity means that during the process of cell division, these genes are less likely to be separated. By observing how frequently traits were co-inherited, geneticists could infer the relative distances between genes. This method allowed them to create more accurate genetic maps, which were crucial for further research. The closer the genes, the more likely they were to be passed down as a unit. This concept was fundamental in understanding the genetic architecture of organisms. Imagine 2 towns located close together on a map. Travelers passing through one town are highly likely to also pass through the other. This analogy helps to visualize how genes close together on a chromosome are inherited together. Similarly, genes in close proximity on a chromosome are more likely to be inherited together. This understanding was pivotal in the early days of genetic research. Conversely, genes located farther apart are more likely to be separated during the process of cell division, much like travelers diverging at a fork in the road. This separation can lead to a greater variety of genetic combinations in offspring. These early mapping efforts, though limited by the technology of the time, laid the foundation for our understanding of gene organization. They provided a starting point for the more detailed and sophisticated maps we have today. They revealed that genes are not randomly scattered throughout the genome, but rather arranged in a specific order along chromosomes. This order is crucial for the proper functioning of genetic processes. This realization was a pivotal step towards deciphering the blueprint of life. It opened the door to modern genetics, allowing scientists to explore the complexities of heredity and genetic variation in unprecedented detail. To create these early maps, geneticists relied on controlled breeding experiments, often using model organisms like fruit flies or mice. These tiny creatures, with their short lifespans and easily observable traits, became the cornerstone of genetic research. By carefully selecting parents with specific traits and analyzing the traits of their offspring, they could deduce the relative positions of genes. Each cross and subsequent generation added a layer of understanding to the genetic puzzle. These experiments, often involving thousands of individuals, were like intricate puzzles, with each generation providing a new piece of information. The meticulous work required patience and precision, as scientists meticulously recorded every detail. The frequency with which traits were inherited together or recombined in new ways provided clues about the distances between genes. This recombination frequency was a key indicator of genetic linkage and distance. Imagine a tapestry woven from threads of different colors, each color representing a specific trait. The intricate patterns formed by these threads told a story of genetic inheritance. By analyzing the patterns in the tapestry, one could infer the order and spacing of the threads, even without seeing the individual strands. This analogy helped scientists visualize the complex relationships between genes. Similarly, geneticists used the inheritance patterns of traits to infer the arrangement of genes on chromosomes. Each discovery brought them closer to understanding the genetic blueprint of life. These early mapping techniques, though laborious and time-consuming, were remarkably successful. The dedication and perseverance of these early geneticists paved the way for future breakthroughs. They led to the creation of the first genetic maps, providing a glimpse into the organization of genes and laying the groundwork for the development of more sophisticated mapping and sequencing technologies. These maps were the first step towards the modern understanding of genetics, a field that continues to evolve and expand. While genetic mapping provided a framework for understanding gene organization, it was the advent of DNA sequencing that truly revolutionized genetics. Sequencing, the ability to determine the precise order of nucleotides within a DNA molecule, provided direct access to the language of life. The development of DNA sequencing in the 1970s marked a paradigm shift in biology. For the first time, scientists could read the genetic code directly, letter by letter. This breakthrough opened up unprecedented opportunities to understand gene function, identify mutations associated with diseases, and trace evolutionary relationships. Imagine finally possessing a dictionary to translate an ancient language. DNA sequencing provided that dictionary for the language of life. It allowed scientists to decipher the genetic code, revealing the instructions encoded within our genes. Early sequencing techniques, though groundbreaking, were laborious and time-consuming. However, they paved the way for the development of faster, more efficient sequencing technologies that have made it possible to sequence entire genomes, including the human genome.